Hello and welcome to this video on how cells lyse and apoptosis happens. Cell lysis and apoptosis are necessary in the human body. In fact, any organism needs these two processes to happen. That is because removing unwanted cells is necessary to prevent them from growing out of control. This is better known as a tumour. On one hand, you want to prevent the removal of excess cells, but you also do not want this to happen without any control. The signals which tell cells to stay alive are called trophic factors and prevent lysis, or it is more accurate to say that they prevent apoptosis, the programmed or expected cell death. Cell lysis is a delicate balancing act that needs a clear-cut initiating step but also barriers to stop it being triggered inappropriately. Cell lysis is a process in which a cell is broken down or destroyed, generally as a result of an external force, or in some cases a condition. It can happen through natural means, for example, infection. On the other side of things, apoptosis tends to be triggered through very specific scenarios, either an intrinsic or extrinsic pathway. Both of these need very specific conditions to be met, and so, as a general rule, apoptosis is far more restrictive than lysis is. Generally speaking, there are many kinds of cell lysis. Some are very specific to mammals like humans, while others focus on single-celled organisms like bacteria. The broad and certainly not exhaustive list of these include cytolysis, which is when cells will break up and practically burst like an overfilled balloon due to osmotic forces that force an imbalance of water inside the cell. Oncolysis, which is the destruction of tumour cells. Plasmolysis, which can happen in plants due to the loss of water, and again, down to osmosis. Immunolysis, which is when particularly erythrocytes, although not always, have hemoglobin release free radicals within them. This then causes them to break up. It's nearly always down to pathogens, and can in most cases be even more specific down to parasitic infections in some instances. There are also situations in which viruses can be the cause of things like this. For example, bacteria are killed quite easily by bacteriophages, which are viruses that specialize in infecting bacteria. They basically use the bacteria up entirely to replicate more bacteriophages, which can then infect more bacteria later. In the process of doing that, they waste them entirely and cause them to eventually break up. One of the ways for this is that the viral particles are released from the bacteria's cell wall, and because of this, they create small holes. These holes allow the movement of water unnecessarily, or in an undesirable manner. This then means that water builds up inside the bacterial cell and it gets destroyed. Something similar but not the same can occur in human cells when they're infected by a virus. The cell membrane can be damaged, and this can cause human cells to undergo lysis as well. Again, for similar reasons but not exactly the same. In other instances, infection with a virus can cause the cell to exhibit certain signals to the immune system. These signals then make it so the immune system understands it's infected with a virus, and it then undergoes either lysis or apoptosis depending on which particular pathway is being acted on. Inflammation can also increase caspase activity. Inflammation generally is tied to infection with various pathogens, and this in turn causes the cell death. Because this is not programmed but generally localized to the site of infection, it's lysis rather than apoptosis. The apoptosis pathway is arguably followed when T cells get involved. These are one of the immune cells and are largely what in your adaptive immune system takes action on your own cells and in some cases pathogens. The T cells come along and they release granules. These granules are an assortment of proteins which are called perforins. As the name might indicate, these create perforations in the cell membrane. As the perforations develop, the cell can no longer maintain its integrity, and it undergoes destruction. Depending on which particular pathway, whether these are a direct insertion, which would be apoptosis, or if they act on a receptor and follow a predefined pathway, it could also be apoptosis. Most of the time, this tends to be under the apoptosis pathway rather than lysis. This is notably important when your immune cells can begin to target foreign pathogens, things like bacterial and viral infections, and they can insert those same perforins and cause cell lysis. Lysing a cell is not only useful in terms of your own body's functions, but in many cases it's an important research tool. For example, getting DNA means you must destroy cells because that's the only way you can extract nucleic material. There is no means by which you can remove it and keep the cell functional. The only cells in the human body that function without DNA are red blood cells, and that's because they have a very simple function and have no organelles that are important for the purposes of getting DNA. Their maintenance is almost entirely self-sustained within them. They last 120 days and then die. They then get replaced. For this reason, every other cell in your body which continues to go on living for a long time and needs to be repaired and maintained has to have DNA, and removing that DNA will destroy the cell. This can be done with water via osmosis, either by having a hyper or hypotonic solution. In the case of hypertonic solution, it will cause water to leave the cell and eventually it will break up. In a hypotonic solution, water will move into the cell and cause it to burst. Neither of these are generally ideal as they tend to be somewhat more time-consuming and difficult. They're also not as easy to control. Often there are other mechanisms used, such as detergents. These help to attack the cell membrane and break it open with much more force than you would otherwise expect to see. Two methods that are commonly used are to add a solid glass bead or to use a sonic device and shake the cells and, in the case of the glass beads, beat them until they break open. These are still technically a form of lysis. Going from lysis, which is a much broader term and covers many different ways that a cell can be destroyed, we can try and figure out the cause and effect, or more accurately, the first cause and last effect when it comes to that. Many of the pathways we've described share commonalities. For example, most of them involve either weakening or directly putting holes in the cell membrane, which allows the lysis to happen. The other side of the equation is when you plan cell death. This is apoptosis. Compared to lysis, which can be activated or facilitated in all the ways described, this system is much better described because there are only two established pathways. These are the Extrinsic Pathway, and they involve things like tumour necrosis factor. The other is the Intrinsic Pathway. It involves mitochondrial stress or DNA damage. In terms of both things like tumour necrosis factor, it has to go through the caspase pathway. It's a stepwise procedure of procaspase 8, caspase 8, procaspase 3, caspase 3, and then apoptosis. There are different extrinsic signals that can initiate these through intermediaries, and in some cases almost directly, as is the case with calcium. It's this extrinsic pathway which is used by T cells when they're trying to remove tumour cells from your body. They will connect with the mutated cell and they will inject a signal. This signal is what starts that caspase pathway. Looking at the intrinsic pathway, such as mitochondrial stress, it's a little different, more so in that it involves something harming the mitochondria or DNA, but it is a good example of how the intrinsic pathway works. The first thing that happens when we talk about the mitochondria is the release of cytochrome C from the mitochondria. This then causes procaspase 9 to turn into caspase 9 and then caspase 3 and 7. These also have their own precursors. There's also something called the APFA1. The creation of this then adds to something called an apoptosome, and then this leads to apoptosis. All of this can be caused by the release of large amounts of either the cytochrome C or calcium from the mitochondria. Both cause the same pathway to come into effect, just one may involve a few more intermediary steps. Both pathways do end in the same way, and that is these two pathways lead to Cas9. Cas9 being a proteolytic enzyme. That is, it targets proteins and breaks them down. In the cell membrane, you can find many proteins, and these provide an important structural role. Without these, you lose structural integrity. No integrity, and the cell wall will break. Whether that is because there are holes in it, it can't resist osmotic forces, the removal of protein has created holes, there are many ways it can happen. As you can see, the cells in your body are kept alive largely by the regulation of a small number of processes and signals. In fact, the caspase enzymes are the same ones used in gene editing to some extent. That may help to understand why they are so tightly regulated for the apoptosis pathways. The lysis pathways tend to involve the more fundamental failures of cell biology. That is, things like osmosis is not regulated, or the integrity of the cell wall or cell membrane has failed, or a pathogen has somehow or other caused something to go wrong. In all cases, lysis tends to rely on something unexpected happening, while apoptosis is planned to some extent. Thank you for watching this video. If you have found it interesting, consider liking, sharing, and subscribing. Please post any comments, questions, or suggestions that you have below.